Module 6, Genetic Analysis. In this short video, we're going to pick up from the design of a genetic study at the level of a gene or genes. One thing to consider as you plan your genetic analyses is how many genes you expect might influence your trait. In the examples given here of obesity, it's been long accepted that obesity is probably a genetic trait in which many genes act together to influence the outcome. It's important to know how many genes you're trying to find as you create and interrogate your study design. One of the theories underlying the possibility of multigenic traits is the possible numbers of multi-locus genotypes and what their distribution looks like. So in this table, it's giving you an example of different instances in which you're considering variable numbers of SNPs, for example, in determining some trait of interest. In the first example, there's only one locus. There's 2 possible alleles and 3 possible genotypes and 3 possible genotype classes. So they can occur with equal frequency. The possibilities can occur. In the second instance, where there's 2 loci, there's locus A and locus B, each of them having 2 alleles, big A and big B, little b. Under genotypes is listed all possible genotypes that can occur with the combination of 2 loci. So there's 9 possible genotypes, and there you see the genotype classes. The genotype classes are created by simply adding up the genotype scores for each individual locus and creating that class. So on the second row, you can see that in terms of genotype classes, there's only one 0 class and there's only one 4 class, but there are 2 ways to make the 3s and 2 ways to make a 1 class and 3 ways to make a 2 class. In the third instance, with 3 loci, an A locus, a B locus, and a C locus, each with 2 alleles, you can see the same patterns repeating. Here you see a graphical representation of the data that you just saw on the previous slide. And in this case, in these graphics, frequency refers to the number of possible ways to create these genotype classes. In the case of one locus, we said that there were 2 alleles and 3 genotypes and 3 genotype classes, and for each of those classes, there's only one way to make them. When you get to 2 loci, already you're starting to see some normalization of the distribution. As we walked through previously, we saw that there was only one way to make the 0 class and one way to make the 4 class, but there were 2 ways to make the 1 and 3 classes and 3 ways to make the 2 class. And as you look over here at 3 loci, again, you start to see that the distribution of possible ways to form those multi-locus genotypes looks kind of normal. This slide extends that analysis to 4, 5, and 6 loci, and these distributions of possible genotype classes is one of the justifications provided for the assumption that quantitative traits that are normally distributed, the genetic component of those traits is likely to be multigenic. In terms of deciding how to focus your genetic analysis, there are 2 main strategies. The first is to look at candidate genes. These are genes that you select based on known or observed function and your hypothesis about how those genes may interact to influence some trait or outcome. But I can also just simply interrogate the entire genome and analyze the variation across the genome in multiple ways. I can march along the genome looking for association to a trait or disease without having any hypothesis a priori about functional genes that I might locate. For example, if I'm doing a candidate gene study for a metabolic outcome, I might focus on genes related to energy and lipid metabolism. I might look at the process of adipogenesis and interrogate those genes. I could look at genes related to satiety signaling or energy expenditure, or I could look at the data from animal models in which a gene has been disrupted and the animal has become obese. And I can take all of the information that has been already reported from genome scans, and I can use that information to guide my selection of candidate genes. And in this case, I focus my analysis directly on the genes rather than on identifying genes through statistical analysis. For example, I might choose to do a candidate gene study by looking at genes in the leptin pathway, which of course we know is a very important pathway in influencing energy balance in multiple ways. So I could choose leptin and the leptin receptor, the JAK and STAT3 pathway, which is the primary way in which leptin influences eating behavior, or I might want to focus on leptin's effects on energetics and look at IRS and PI3 kinase and AKT and mTOR. And I could choose all of those candidate genes, and I can select them and focus initially and directly on a hypothesized pathway. So once I've identified my candidate gene, the next question is, which SNPs am I going to interrogate in those genes? And as you know very well at this point, there are many databases that I can go to identify those variants and choose them directly for interrogation. Of course, GeneCards is super helpful. The NCBI has many resources, UCSC, the Human Gene Mutation Database, and many other places where I might go to identify the SNPs that I want to analyze and genotype in my study. When I do those analyses, I can use very straightforward analytical strategies, and these are things that you're very familiar with. In the case of analysis of variance or regression, I'm simply looking at the distribution of some quantitative trait by a genotype. I can also use things like chi-square to look at the frequencies and proportions of a trait or outcome, obese versus non-obese, and a particular genotype. And I can use these types of analyses. I can use covariates if I think it's important, and I can look at the effects directly of a particular SNP or variant in the genome on an outcome of interest. Another way you might look at the effects of genotype is by looking at survival. And in the case of survival analysis, it doesn't necessarily mean just literally surviving. It can also represent time to an event. So in this graphic, the a genotype is associated with earlier time to event, which the event could be death, but the event could also be something like the formation of a tumor or the time to remission. So it can be different kinds of events, and you can do those types of analyses looking at the effects of genotypes. In terms of statistical analyses, you want to make sure that whatever you do, it's appropriate for the study design you've selected, it's appropriate for the subject sample, and that you've taken into account uniqueness and particular traits in your subject sample, and that the statistical analysis is appropriate to answer your research question. So there are very complicated and complex ways of looking at genetic data that goes beyond the scope of this class, but it's important that your analysis match your study design. In this case, A linkage analysis might be used to look at segregation in families, and an association type of analysis might be used to look at cases and controls or cohort kinds of studies. The probability of finding a gene for a complex trait has been likened to looking for not just one set of encyclopedias, but a whole set of sets of encyclopedias for the volume, the page, the paragraph, and the line that contains a single character defect. And you can imagine that that whole task is extremely difficult if I was only had at my disposal a bunch of books and the random process of picking a page and a character and testing its effect. So we already talked about candidate gene studies, which are based on hypotheses about function. And now we can use other types of strategies to look across the genome. The process of mapping a gene is the same sort of process that you might use if you were trying to place some interesting location on a physical map. If I asked you to find some interesting place and told you it was somewhere in the state of Texas, even though Texas is smaller than the world, it's still a pretty big area to search. And that would be similar to me saying, find some interesting gene and it's somewhere in the human genome. But if I told you that that interesting place was in Houston, Texas, that would narrow your search quite a lot. And that would be similar to me saying, look on chromosome 6, for example. Now, Houston is a big place too. But what if I said the interesting place that you're trying to find is somewhere inside loop 610? That would narrow down the spread of your search quite a lot, although it's still a big place, and that would be similar to me saying something like look on the P arm or the Q arm of chromosome 6. And now if I told you that this interesting place was somewhere between downtown and Memorial Park, you could drive back and forth between those two places and you're likely to find Houston's own beer can house. The man standing in front of this house drank a lot of beer, and every time he did, he took the cans and he either smashed them or he cut them and covered his house. And this lady standing in front of this house quickly sold the house to the orange show after her husband died and moved into some place that wasn't covered with beer cans. So driving back and forth until I find my interesting place is similar to taking landmarks along the genome and basically running back and forth and testing the effects of those landmarks one by one. In linkage analysis, I'm taking advantage of family structures and known probabilities of allele sharing between different types of family members to identify signals from places in that genome. Deviations from expectation of allele sharing between family members then can be used to identify genes that are related to a trait using linkage analysis. For example, the probability that monozygotic twins share the same genes is 100%, as we said before. And the probability that dizygotic twins or full siblings share the same genotype can be calculated as follows. The probability that any one offspring receives an A1 allele from either parent is 1/2. And the joint probability that both siblings share the A1 allele is 1/2 times 1/2, or 1/4. The combined probability that both sibs share the same 2 alleles is 1/4 plus 1/4, or 1/2. So we know that the probability of any two sibs sharing the same genotype is on average about 1/2, and the genetic correlation between two full sibs is 0.5. In families, basically what you do is you track the segregation of a particular genotype through the pedigree, looking for places in the genome that are always present when the trait or the disease of interest is present. So if you notice here at the top, there's an affected mom, and she has this little red mark, which is supposed to represent the affected, the locus that drives the trait of interest. In the next generation, all of the genotypes are going to have a green allele from dad, and they can have some part of the yellow allele from mom. And as you see, that part of the yellow allele that was colored red is segregating with the person in the second generation that is also affected. Now she is marrying her husband, and he has two orange alleles. Now in their offspring, their children can only receive orange alleles from dad, and from mom they can receive green alleles or yellow alleles with the red part. And of course, you learned earlier that in the formation of eggs and sperm, the chromosomes align and crossing over takes place. So mom's chromosome is going to be a mix of her two parental chromosomes, and you can see that in her children, you see this mix of chromosomes, but that part of the chromosome that's colored red is always segregating with those two individuals that are also affected. This affected woman marries her husband with two pink alleles, and their children have to have, each of them, one pink allele from dad, and some combination of mom's recombined alleles. And in this example, the recombination is a little bit exaggerated. But again, you see that those individuals that are affected always have that genotype represented by the red color. And in this example, you can see that the red color is segregating with the trait or disease, and much more often than you'd expect by chance, which is how you identify places in the genome that might be encoding a trait or a disease. In association analysis, you might be looking at cases and controls, and you might have an hypothesis that says affected individuals will share an allele at a higher frequency than what's seen in unaffected individuals. And so you might do that analysis of variance or that regression analysis, looking at genotypes and comparing genotype frequencies between affected and unaffected. In the case of genetic analysis, what's important to understand is that cases and controls are actually part of a larger population that is probably related at some level. Populations have common descendants, and this is why we need to be careful about how we choose our cases and how we choose our controls, and whether or not there's some substructure underneath the cases and controls and the proportions of cases and controls. So important concepts in genetics to consider are things like penetrance. How frequently and how robustly is a trait expressed given that the genotype driving the trait is also present? And that can be variable. Anticipation refers to the effect of genotypes that occur with earlier and more frequent onset. And random versus assortative mating, which we've talked about earlier, refers to the fact that as assortative mating happens and people come together who are affected, the likelihood of seeing the affected state in their offspring becomes greater. Genome-wide association studies basically take these markers that are known along the genome, and the more we've done genome-wide association studies, the more densely we've been able to interrogate the genome. Basically, each of those markers is a SNP or some kind of variation that's known, and we can test them by doing the same kinds of analysis we talked about earlier, by looking at genotype frequencies, for example, in cases and control, or by looking at the distribution of some quantitative trait by genotype. We basically do those analyses over and over again, looking for signals in the genome. Obviously, if you do enough analyses, some of them are going to be significant just by chance. So it's important in genome-wide association studies that we control for the number of tests we've done. And of course, we know that genes don't act independently, that genes and environments interact to influence an outcome of interest in all kinds of ways. And that could be behaviors, it could be nutritional environments and physical environments, exposures to toxins and drugs and other factors that we know influence outcomes and diseases and metabolism. And so we want to consider, when possible, environments that may influence the effect of genes. In that case, we may go from the simple analysis of variance strategy to doing a stratified analysis in which we repeat the analysis but looking at the effects within a given environment. For example, environment 1 may be a low-fat diet and environment 2 may be a high-fat diet, and you may see differential effects of genotype given that environment. So geneticists always refer to anything that's not genetic as an environment, in case that example sounded a little funny to you. Similar to association studies using analysis of variance or regression, we can also take our survival analysis and do a stratified analysis there, in which we're looking at survival by genotype within 2 different environments, exactly as we did before. So in this case, you can see 2 very different patterns of survival or time to event based on whether or not the individuals are ascertained in a given environment. We've said before that many complex traits are likely to be driven by multiple genes. So how do we test the combined effect of multiple loci? This is an example from some of our own work in which we examined 13 SNPs in the FTO gene for their association to exercise dropout. In this case, a very simple chi-square analysis was done across these 13 variants to see whether or not the frequency of genotypes in these loci was associated with yes, no, whether or not the subjects dropped out of exercise. Of the 13 SNPs, 5 of them showed a significant signal increasing risk for exercise dropout. Now, this is the original SNP that was identified in FTO as being associated with body mass index. And there are many, many variants in FTO, and it's a really big gene. So there isn't anything really obvious that would say why we might see this effect in these particular SNPs. So we want to go forward and develop a strategy for looking at the SNPs in a multivariate way. One of the things we can do is simply conduct multivariate regression or multivariate analysis of variance. But the problem with that approach is that we pay a cost in terms of degrees of freedom each time we add an additional variable to the model. And you can imagine there are only 5 significant loci here, but if I have way more than 5 because I'm interrogating the genome very densely, doing a multivariate analysis in which I just simply put all the genes in the model at the same time would be a tremendous price in terms of lowering my ability, lowering my power to detect a significant effect. One strategy that's been used to examine the joint effect of multiple loci is to assign a SNP score to the genotype at each of the loci, and then add the scores to create a genetic risk score. For example, in this case, here are the 5 variants that were associated with exercise dropout, and in the 2 column represents the risk-raising alleles, the alleles at these loci associated with increased dropout. A genetic risk score is created by simply adding up the scores at all of the loci that are being included. So, for example, a person who was heterozygous at all the loci, at all 5 loci, would have a genetic risk score of 5, 1 point for each risk-raising allele. And various combinations of genotypes would have a genetic risk score ranging from 0 to 10. This quantitative measure can then be used in regression analysis or analysis of variance, depending upon the distribution of the genetic risk score and how many loci are included in that score. In the case of FTO, any 3 alleles, after calculating the genetic risk score, any 3 alleles are associated with more than twice the probability of dropping out from exercise. This was a really powerful way of detecting a joint effect in the FTO gene. In fact, if you look at the distribution of risk-raising alleles and you correlate that with the quantitative measure of exercise tolerance, you see this dose effect of the risk-raising alleles in being associated with increasing number of alleles, being associated with lower exercise tolerance. So in this example, a genetic risk score turns out to be a powerful way to look at the combined effect of multiple loci. This little table kind of summarizes the strategies that you might use for analysis based on your type of study design and based on the trait or disease that you're interested in. So for example, in terms of family-based studies or linkage analysis, if the trait is discrete, as in the example that we showed, you could look for tests of deviation in expected allele sharing. If the trait is continuous and it's a family-based study, there are regression strategies for looking at expectations of allele sharing against a distribution of a quantitative trait. We didn't go very deeply into the types of more detailed linkage analyses, but there are strategies for looking at those kinds of traits in families. In terms of population-based association studies, we talked about chi-square and Fisher's exact. We can use logistic regression to look at outcomes and other types of regression-based models. And certainly for continuous traits, we can use regression and analysis of variance to test the effects of genetic variation on quantitative traits. Once you've identified some place in the genome that's possibly containing a gene that influences your trait of interest, there are many different strategies for functional tests that you might run to see if you really have identified the pathophysiology of your disease or the physiology of your trait. Things that will identify the functional variant. You can look at things like gene expression or protein levels or protein binding, all kinds of functional assays using some of the technologies that you've already heard about as a last necessary step in showing that your statistical finding is actually a functional finding as well. And once you've done these amazing studies and you identified a functional effect, It's time to take your Nobel Prize.